which is nothing but a covalent chemical bond which is formed between two amino acids with a free amino group and a carboxyl group. Everybody knows about that. Everybody knows about simplest amino acid glycine. But the peptide complexity starts from here. We are not dealing with the structure glycine, benzene. We are dealing with the structure where glycine or halogenated glycine or amino acids are just smaller units of the complex structures. So here some of the modified amino acids you can see: isoserine, DHHA, beta hydroxy, parabromophenylalanine. See, it is an antifungal cyclic deoxypeptide, cyclolistid A. Here two unique amino acid units are found: formyl leucine, chloroisoleucine. These are the part. Next structures I want to share with you: persipeptides A and B, N-methylvaline, N-methylphenylalanine. N-methylation parts are there. Usually the bioutility is N-methylation modulates biological, structural and pharmacokinetic properties of the peptides. And a lot of research is going on this particular concept. Scientists are synthesizing N-methylated peptides and they are lot bioactive. Some peptide with the unique acidic moieties: thalsosparamide G with the gamma amino acid units like AHPEA and ADPA. The respective example is cited. The cyclic peptides involving heterocyclic units like isoxazole, then thiazole. We have seen in the heterocyclic chemistry mostly this nucleus, but these nucleus as a part of a complex structures are rare. Some other moieties are involved like dihydrohomohelanin, bromotryptophan, isoserine. Respective examples are cited with the cyclic structures. Whenever we are talking about the peptides, we may face linear moieties as well as the cyclic moieties. See some of the examples I cited: TRH, thyrotropic hormone, which is a linear peptide. Then longicalicin A, you can see it is a cyclic peptide. It is a mixture of a cyclic and a linear peptide. Whenever we are comparing cyclic and the linear peptides, the cyclic are usually preferred over the linear one due to the two main reasons. Inherent flexibility of linear peptides lead to different conformations which can bind to more than one receptor molecule, resulting in undesirable side effect. I think all are well aware of the lock and key mechanism of the enzyme theory. One lock, one key easily opening, means one receptor, one attachment, proper biological response. Otherwise there will be variable response. Another factor is cyclization of peptide reduces the degree of freedom for each constituent within the ring, and thus it leads to reduced flexibility, increased potency and selectivity of the cyclic peptides. Some of the isolation sources: marine sponges are the major sources for the cyclic peptide. Jaspis species, some of the pictures. The marine mollusks, cyanobacteria, hypnocytes, fungi, bacteria, plants. Bioactive of peptides: cytotoxic activity against various cell lines. Some of the examples of the cyclic peptides are cited: cyclooxazoline, stylostatin, discoquinonides, discodermins. With antifungal activity: jasplaquinolide, hymenamides, aceclutins. Anti-malarial activity: carbamazine, dragomaline. Some of the other activities are associated with the cyclic peptides with the respective examples. Mechanism of action: two major activities, cytotoxicity and antifungals. Two mechanisms were found to be involved. First one, for the cytotoxicity, apoptosis, that is nothing but a programmed cell death. Here the inhibition of tyrosinase phosphorylated IFG1 receptor is involved. With the antifungal mechanism, inhibition of enzyme beta 1,3-D glucan synthase is involved. Some of the cyclopeptides in clinical trials with the anti-tumor activity: RAV, RA7, then aplidin, with the immunosuppressive action, astin C. Some of the cyclopeptides which are currently used in the clinics: bacitracin for topical administration, polymyxin B with topical, gramicidin as topical, vancomycin IV and oral route, daptomycin IV. If we are talking about synthesis, two general techniques are there: solid phase, solution phase. So our research team worked 10 to 15 years on the solution phase synthesis, and we have published near about 25 papers in international journals and got recognition about the fact. So we synthesize cyclotetrapeptide, cyclomontanin D, corvide heptapeptide B, cyclotetrapeptide, gypsum D, and so on. So here, just to demonstrate example how we carried out the research work. Before that, we should go for the protection and the deprotection of free amino and carboxyl group, of which the whole solution phase synthesis is based. So a free carboxyl group and free amino group should be protected whenever the two amino acids prior to coupling of those. So amino group can be protected by BOC group, tertiary butyl oxycarboyl group, and carboxyl group can be protected by esterification. And similarly, deprotection of the BOC group is feasible with TFA, trifluoroacetic acid, and deprotection of the carboxyl group is possible by alkaline hydrolysis with lithium hydroxide. So here I demonstrated one of the example how gypsum D, which is cycloheptapeptide in chemically, can be synthesized. So first we have to synthesize a tri and tetrapeptide units, which will be coupled in the later phase to a linear heptapeptide unit, and finally it will be cyclized to form the natural molecule, which will be identical in synthetic aspects. So this is the final cyclization step. Here a linear heptapeptide unit is got cyclized into cycloheptapeptide unit, which is identical in structure to the gypsum D. Controlled delivery of peptide proteins. Obviously there is need of controlled delivery because orally instability issues are there. So it can be solved by delivery through microspheres, microcapsules, nanocapsules, implants, hydrogels. So some of the material which can be used to prepare microsphere for controlled delivery are starch, alginate, chitin, then chitosan. Some of the method used for the preparation of nanocapsules: polymers, most commonly polymer used are polylactoglycolic acid, polylactic acid with the respective drugs which are peptide in nature. Some of the marketed formulation of proteins based upon biodegradable microspheres: leuprolide acetate, recombinant human growth hormones with the respective diseases like prostate cancer and GH deficiency. Advantages of controlled drug delivery, controlled delivery of peptides are: it can provide maximum stability, activity, and bioavailability. Moreover, it provides improved efficiency, reduced toxicity, and improved patient compliance. Some of the examples I have cited for the peptides and proteins in clinical use or undergoing clinical trials. So everybody of us is aware of tissue necrosis factor, proleucin, but chemically these are peptides which are in the body. Fibroblast growth factor, insulin-like growth factors. Some of the bibliography I cited. Thanks a lot.
Thank you. Thank you. Any question? Dr. Dahiya. We have next speaker, and today's last speaker is Dr. Saurabh Dahiya. He is from APJ Staya University, India, and he is going to speak on Guar Gum Revisited, potential carrier for targeted drug delivery systems.